Again, we're moving on to cells. We've skipped over a little bit of chemistry, but I think that's probably a good way to go, and that's the way they do it up north. So cells are the basic units of life. They're units of structure and function in the body. Anything that happens in a body, anything at all that you can think of, has got its basis within the chemistry that's going on inside of a cell of some type. Cells are incredibly small. Most of them are not visible without the microscope, and they weren't discovered for a long time because of that. In fact, they're on the order of magnitude of micrometers, which we call microns. Each individual cell that we'll talk about is specialized characteristics. They're parts of the body, and they have all been differentiated. So an individual cell within, or most individual cells within a multicellular organism, is not going to be able to reproduce an entire organism. It's just too specialized. It's gone through too many changes. It's designed to do some very narrow but specific functions for a cell. There are millions of different kinds of cells in the body. I don't know, millions might be too many, but there are a large number of them, and they are varied in their size and shape, their structure and their function. They are all interrelated. They work with one another. They need one another. And today we're going to look at some general aspects. We'll talk about what we call a generalized cell. It's got things that most cells will have, and when cells have them, they will do what we say they do today. But there is really no generalized typical cell that we can look at. We can go all the way from nerve cells, which are incredibly long but narrow, to sheet-like cells we'll find in epithelial tissues when we talk about tissues next week. Muscle cells are organized with contractile proteins so that they can get forcibly shorter. So a composite cell or a generalized cell doesn't really exist, but it's a way for us to start learning about cells. It has the components we'll find in almost all cells. Anything we actually call a cell will have these three components. A nucleus, usually in the center, that's surrounded by cytoplasm, and it's all held together with an outside membrane called the cell membrane. And actually, I would begin, if I were you, to refer to it as a plasma membrane, because any membrane within a cell is technically a cell membrane. Within the cytoplasm, we will find all kinds of organelles. We can't see those with a compound light microscope, but we know they're there from other methods. They will have specialized functions, and the fluid around those organelles in the nucleus is called cytoplasm or cytosol. So here is a composite cell. It's got a lot of parts from many different cells. You rarely find flagellum and cilia on the same cell, or microvilli and cilia on the same cell. But these are things you're going to run into as you're going through typical cells in the body. The outer part of all cells is called the plasma membrane. It maintains the integrity of the cell. It does so because it has the ability to be selectively permeable. It regulates what can get in and what can get out of the cell. Sometimes you may hear me slip up and say it is semi-permeable, and the basic membrane is semi-permeable. But the plasma membrane is more than just semi-permeable. It is selectively permeable. It really picks and chooses what goes in and out. It's also the location for a lot of signal transduction. Information from outside the cell gets in via receptors on the cell plasma membrane. And when it wants to talk to other cells, those materials that it talks with have to be released through plasma membrane. It's mostly lipids in the form of phospholipids. There are also proteins in there. And on the outside of the cell surface, there are some carbohydrates, and many of those carbohydrates are used for identification information. It's a phospholipid bilayer because those phospholipids are a special kind of molecule that is got two major parts to it. A water-soluble part, that's the phospho part. It likes to be in water. And two lipid tails, the hydrophobic parts, that do not like to be in water. They organize in two layers so that the one layer of lipids will face below it another layer of lipids, and then on the top and the bottom will be the phosphate group. So you can think of it as a sandwich with phosphates, lipids, phosphates. Phosphates being the bread, the lipids being the peanut butter, if we're going to call it a peanut butter sandwich. It is permeable to some things. Lipid-soluble substances can get through the plasma membrane. So in that sense, it is semi-permeable. It doesn't block everything. But water-soluble substances and water itself, for the most part, cannot get through the phospholipid bilayer. That phospholipid bilayer makes up the bulk of the barrier between the inside and the outside world. Within those lipid tails, there are cholesterol molecules that help to stabilize the membrane. They increase its impermeability to water-soluble substances, but they also increase the fluidity of the membrane. We have to think of the membrane as behaving like a fluid. Things move left to right inside the membrane. They don't flip. They don't go from outside the cell to inside the cell, but they do move from left to right in a fluid way. So we call it a barrier, but it's a very fluid barrier. And in fact, the model we use to describe the plasma membrane is called the fluid mosaic model. It's those mosaic, because there are a number of different things in it, are moving freely within the membrane. There are all kinds of membrane proteins. When we talked about them in biology, simplified that they were transport proteins. They are not all transport proteins. Many of them are. There are some pores. There are some channels. There are receptors. There are enzymes. There are contact identification. And the important class of molecules we find in membranes, especially those cells that crawl or are used for crawling over, are called CAMs, which are cell adhesion molecules. And again, those carbohydrates tend to be recognition molecules, self-markers. When you give a transplant, you check those carbohydrates to see if it's when you say a close match between individuals, it's those carbohydrates you're looking at for a close match. So if we looked at cell membrane structure really closely, we would see those heads of phosphate groups in a layer of lipid tails organized in this way to give us the bilayer. So we see there's one layer and there's the other layer, and the way they're organized, that is a barrier between the outside world. Within that barrier, if we look at it from a slightly different angle, we've got transmembrane proteins that go all the way across. We've got integral proteins that are embedded within the membrane and may not go all the way through. There are also peripheral proteins that are associated with one side of the membrane or the other. Embedded within here in the phospholipids, in the lipid part of the phospholipids, there's cholesterol molecules. Here's transport protein that allow a channel coming through. And then stuck to mostly the outside, we have these carbohydrates attached to either lipids. So here it's attached to a lipid, so it's called a glycolipid, and here it's attached to a protein, it's called a glycoprotein. Those are the carbohydrates that we use for a cell identification. So we can have problems with ion channels. If you have a mutation in sodium channel, that can cause the inability to feel pain or extreme pain conditions, and that is a very dangerous condition. It's a very rare condition, but people who don't feel pain don't know when to quit what they're doing, and they usually end up dying by misadventure. Mutations in potassium channels can disrupt the electrical activity. Potassium is particularly important in the heart rhythms, and it also is involved in hearing, as well as several other nervous system functions. Abnormal chlorine channels are the cause of cystic fibrosis, and cystic fibrosis is a common genetic disorder that leads to the production of thick mucus, which causes difficulty breathing. It also has issues going on in the pancreas. It's often used to be, at least before testing, early detected by the salty taste of the sweat of the baby. And there are other problems that come with cystic fibrosis. And although the treatment of cystic fibrosis has improved greatly over the last 30 years. So those cell adhesion molecules are molecules that help direct cells where to go, cells that are crawling in particular. They're going to be very important in immune cells. There are a couple of classes of those are selectins. Selectins are on the surface of white blood cells, and they anchor the white blood cells where they're going, giving some friction, kind of like grabbing a hold with a handhold. And then there are integrins. The integrins are on the other cells, capillary cells, for example, and they are going to, when they're expressed, they'll provide something for the selectins to grab a hold of. Integrins are also very important in embryonic cells, and we don't talk a lot about developmental biology in here, but there's a lot of cell migration that goes with development of an embryo, and that requires a lot of integrins, selectins, and other classes of molecules that do similar things. In the cytoplasm, we've got a network of membranes, organelles that are suspended in there. So the cytoplasm is the cytosol plus the organelles. According to this book, so the cytosol would be the fluid portion of the cytoplasm. Organelles are tiny structures with specific functions that we'll be going through here shortly. The cytoplasm contains a cytoskeleton, which is a framework that provides the three-dimensional structure to the cells. It's awfully easy to think of a cell as very small, very thin, with no real three-dimensional structure, but it does have very important three-dimensional structure. So those organelles include ribosomes. Ribosomes are composed of proteins and RNA. They're unusual for that reason. Most things we'll talk about in the cell are composed of proteins. They have some that are free in the cytoplasm and some that are attached to endoplasmic reticulum, giving you rough endoplasmic reticulum. They are going to provide structural support during the process of building a protein in the cell. They help the messenger, they hold the messenger RNA so that it can be translated by transfer RNA. Endoplasmic reticulum is a series of membrane-bound sacs, canals, and vesicles. It's a tubular transport system. There are two kinds of endoplasmic reticulum. Rough endoplasmic reticulum, which has ribosomes attached to it. That's where proteins are being made into this transport system. So ribosomes are on the outside edge, building the protein inside the transport system where other things are going to happen to those proteins. And most proteins in the cell make their way are built into the endoplasmic reticulum and then delivered where they need to go. There is also smooth endoplasmic reticulum. Smooth endoplasmic reticulum is involved in the manufacture of lipids, also involved in some detoxification. Not all cells have a lot of smooth endoplasmic reticulum, but when we get to the liver, you'll see that it does have a lot of smooth endoplasmic reticulum because of its function. Vesicles are membranous sacs that are used to store and or transport substances when things are moving around. Beyond the endoplasmic reticulum, we come to the Golgi apparatus, which is again sacs, but these sacs are more flattened. Here we refine what happened to the protein, package them up to where they need to be delivered to, and send them on their way. Could be to the surface, cell membrane, could be back to the nucleus, could be to another organelle, but they're packaging and distribution centers. Milk secretion, is an example of an organelle interaction in the transport of substances by vesicles. So milk is synthesized in the smooth endoplasmic reticulum, makes its way, the lipids are from the smooth endoplasmic reticulum. The proteins are synthesized within the rough endoplasmic reticulum. They join together in the Golgi apparatus, and they are secreted out as droplets. Mitochondria are membrane-bound fluid sacs where the chemical reactions that make ATP occur. Cellular respiration, I'm sure you all remember that. The pyruvate makes its way into the mitochondria, where it goes through the matrix. The Krebs cycle and electron transport system are housed there, which is why mitochondria are called the powerhouse of the cell. Lysosomes are small sacs that have enzymes that digest proteins. Some digest carbohydrates, nucleic acids, bacteria, debris, worn out parts. They call it garbage disposals here. I would think of them more as a recycling organelle, because those materials that we're breaking down are going to be reused. There are also a similar class of organelles called peroxisomes, which have enzymes that digest things like lipids, alcohol, and hydrogen peroxide. So their targets are a little different. Microfilaments, microtubules, and intermediate filaments are structures that make up found in the cytoplasm that make up the cytoskeleton together. The microfilaments are the smallest. They're tiny rods of actin. They provide for cellular movement if that is needed. And they're used as muscle contracting proteins. We'll see them again when we get to muscle. Microtubules are large tubes of tubulin. They're in the bigger. They have rigidity that maintains the cell's shape. They're also involved in cilia, flagella, and in the centrioles when we get to cell division. And they're important for moving organelles around. Intermediate filaments are composed of several proteins, and they provide a cytoskeletal structure, and they are also important in the nuclear envelope. Centrosome is a structure that's made up of two centrioles. It's in the cytoplasm near the nucleus. It's used for spindle fiber formation during cell division, and that helps to distribute the chromosomes equally between the two daughter cells. Cilia are extensions on the cell membrane. They are microtubules in a cylindrical pattern, forming a fringe on epithelial cells. They're shorter than flagella and very abundant when they are present. They beat back and forth in a rhythmic fashion, a coordinated fashion, to move things along different places in our bodies. For instance, they are on the respiratory tract, and they move mucus from the lower parts of the respiratory system to higher parts of the respiratory system. They are also used to propel the egg toward the uterus. Flagella is an extension from the cell membrane. There's only one in humans, and only about 50% of the population has it. It's found on a sperm, and in humans there is only one flagella. There are some organisms with more than one flagella, but humans' sperm has one flagellum. Milase is a mutant gene in the DNA of mitochondria, and that person with that mutation can't get the maximum energy from nutrients, can't generate all 36 ATPs from a glucose. Krabbe disease is caused by an inability to produce one lysosomal enzyme, and that person cannot produce myelin for nerve cells, and that can lead to severe nerve damage in the nervous system. ADL is adrenoleukodystrophy. It's caused by a lack of a protein in the membrane of peroxisomes, and that leads to fatty acid buildup, destroying myelin sheaths and nerve cells. And when that occurs, the nerves can't transmit impulses fast enough. Next, we'll look at the nucleus. That contains the genetic material, controls cell activities. We have nuclear pores within that membrane that are separating the nucleoplasm from the cytoplasm. Excuse me. Those nuclear pores allow the passages of certain substances in and out of the nucleus, making the nuclear membrane, again, selectively permeable. The nucleolus is a dense body of RNA and proteins. It's the place where ribosomes are being built. And chromatin is the DNA, but not only the DNA, the proteins that give it structure that are controlling it. It is where information is stored for protein synthesis. And I will